Thank you so much for the introduction. Sorry for any inconvenience that has been caused for missing my slide. And I know there is a lunch break followed by my presentation. I will sum up the presentation in short words. Hope it will be quick. As you see the title, so we want to see whether the drinking water quality of poultry impacts the course of infection and vaccination we do. And the data I'm going to present comes from controlled studies. It's all about controlled studies. Okay, so I'll start with a very powerful quote which says that water is life and clean water means health. And a fact is that poultry on average drinks twice as much water as they consume feed. So some of constituent of concern in water could be bicarbonates, carbonates, nitrates, sulfates, iron, sulfur, and as well as microorganisms like E. coli, Salmonella, and Pseudomonas. And these contaminations can come from either naturally in the water or that they can come from the sewage, animal waste, or dead animals can contaminate the water. And now I move to the significance of IBV, infectious bronchitis virus. It is endemic in North America and only in Canada, it causes more than $200 million loss each year. And it has been shown that contaminated water as a link to suppressing immune system of chickens. And only in Alberta, different strain of IBV exist in North America. There's a strain like Mars type strain. It leads to 40% reduction in egg production in years. And the gap we saw is there is limited research how poor water quality influences the viral replication and vaccine efficacy in layers. So selection of the water, it's a huge project with multiple objectives. One objective was to collect the sample from different regions in Alberta and analyze the water samples based on its physical, chemical, and microbiological parameters. And then what we did, we developed a scoring criteria and used a ranking system to rank the water from best water to the worst water. And the highest scoring means the poorest water that in my following presentation, I will term as feed water was selected for these controlled studies. So how the experimental design goes, we did two experiments and both are controlled experiment. One with the infection, the other one with the vaccine. What we did, we took 48 specific pathogen-free chickens, divided them equally into two groups, 24 in each group. One kept on the tape water, which is our controlled water, and the other one on the field water that we ranked the poor water. And then again divided each group into two, as you see in the slide. One group as a control, the other infected with IBV. Similarly, in the field water, we made two further groups, control and infected. Now I will jump to the result. What we saw interestingly in the outcome was the clinical sign. It should be in both groups because both are infected. But what we saw, the field water group became more sick. They were severely sick compared to the tape water group, as you see from the graph. And we followed our samples. We saw the field water birds are shedding the virus more compared to the tape water group. It means the virus is replicating at higher rate in the field water group compared to the tape water group. And again, you see in the square, at 4 days post-infection, we saw that viral genome loads in different tissues like trachea, lungs, and cecal tonsil. In all of these tissues, the viral load is higher in the field water group. And we also looked at the histopathology. Due to the time shortage, I will not go into the detail, but you see more severe damage in the tissues at the far right. It is trachea and beneath is lungs. They are damaged at higher rate compared to the tape water groups. And these are the lesion score. We also score the histopathology lesions. And again, you see the orange bars higher compared to the gray bars. So the orange bar is the field water group. And we wanted to know why the I virus is replicating. Then we moved and we wanted to see the cell-mediated immune response. We saw suppressed CD4 cells, which are helper cells, and also lower level of CD8 cells, which are the primary cells responsible for virus clearance from the tissue. So the cells in the lungs are suppressed. We noticed lower population of CD8 cells. And in experiment 2, we had the same design that I showed you in an infection. It is the only difference is instead of the infectious virus, we used vaccine, which is much relevant to the field data. And what we saw interestingly, there was 600 units of less antibody titers in the field water group, which when it comes to IBV, which is a huge amount of titers, and although they're kept in the same condition, but the field water group had less titer compared to the tape water group. And is in accordance with our infection data, the CD4 and CD8 cell were again less compared to the tape water in the field water group. So to conclude from our two studies, that we found that field water impaired development of immune system, lower the percent of CD4 and CD8 cells in lungs, which are actually responsible for viral clearance and tissue protection. We also found the field water group had higher viral genome loads and severe tissue damage. The antibody response was also weak in the field water group. So we suggest that there is immunosuppressive effect of suboptimal drinking water quality in poultry. For future directions, we suggest that we should repeat the trials with other viruses and vaccines. And we also should focus on other tissues like kidney and thymus, which are also immune organs. And we want to conduct the same trial. We kept these birds only for 7 weeks, but we should keep them for extended period of time till they come in production and see how that quality affects the production in birds. And these are the references. With that, I would like to thank my supervisor, Dr. Kareem, who is here attending conference with me from University of Calgary, all members of Kareem Lab at Calgary, Gomez Lab at Saskatchewan, egg farmers of Alberta, and prairie livestock veterinarians who help us in sampling and recruitment of the farmers, and Marshall Swine and Poultry Health Institute, and Sylvia and Karen, who are professors at University of Calgary. Among these, I would also like to thank all the funders of these projects. And thank you so much.